What do you think, Rafa, Tina? Should we get started?
Yeah, we can go ahead, Doug.
Oh, there's a few more coming in. 64. Excellent. All right. Well, we'll start kicking things off. Welcome to today's off-season local AIS partner workshop online. And today we're going to focus on detection, response, and management. If we've not already met, I'm Doug Jensen. I'm a DNR's AIS prevention planner. I'm one of your co-hosts today, along with Rafa Contreras-Rangel, who replaced Tina Fitzgerald last summer as our Southern AIS prevention planner. Rafa, would you like to introduce yourself?
Yeah, as Doug said, the new AIS prevention planner for the southern counties. My name is Rafael Contreras Rangel, but you can just call me Rafa. It's much easier. And before this, I was the AIS e-commerce prevention planner. So some of you know me with some work that I was doing with that. And yeah, this year I'm just excited to get to know the position and get to know everything you all are doing. And yeah, I'm just helping out with these workshops for the first time. So thank you.
Thanks, Rafa. And Tina's also been helping out with our workshops. Tina, would you like to say hi?
Afternoon, everyone. Tina Fitzgerald. I'm the new Watercraft Inspection Program Supervisor, but previously held Rafa's role as the AIS planner. So just happy to host these workshops again. It's our favorite thing to do. So happy to help out. Nice to see everybody.
Thanks, Tina. And thank you all again for joining us today and participating. First, we're just going to go over some workshop goals. So for the workshops, we always organize to make sure that we're all learning from each other's experiences and networking with your peers. We do want to set expectations for our workshop today as well. We want everyone to contribute to the conversation, be active listeners, speak honestly, and to share information in ways that can help others be successful, be respectful and open to new ideas, new ways of doing things, and make sure that everyone participates and nobody dominates. We'd like to take a moment to remind ourselves that what we're really working on here is the big picture for Minnesota, and that's including working with the DNR, working with our local partners, along also with your partners, including federal, state, and tribal agencies, businesses, and nonprofits, and many of the lake associations. And our goals are to prevent the introduction, contain the spread of existing, and reduce the impacts of aquatic invasive species in our water resources, for recreation and also communities that depend upon healthy waters for jobs in our economy. And to reach those goals, we in part rely upon really common sense, but we also remain open to new and innovative approaches to help us improve management all across our county, statewide, and beyond. And that's one of the main purposes of this workshop today. So we'll be hearing all about those during our 3 workshops online. Before we begin, just a reminder that today is the first in a series of workshops with this one, another one on partnerships on February 20th from 9 to noon. Also public engagement February 25th from 1 to 3. You should have received from Rafa invitations to all 3 of the workshops. And then we're going to take our chances if the weather holds, and we'll hold some in-person meetings. One in the south at the Chanhassen Library on March 11th from 10 to 3, and March 12th or 13th. We haven't determined a date yet, and that'll be a backup for poor weather. And then for the north, we'll have one in Brainerd at the Crow Wing County Courthouse Building, March 31st from 10 to 3, with our April 2nd as our backup in case of poor weather. So with those introductions, I want to make sure that everybody has been introducing themselves during in the chat. We'd like you to please type in your name, your affiliation, a location, how long you've been in your position, along with an answer to our icebreaker. What are you looking forward to in 2025? We'd, of course, preferred to have you do this all unmute and introduce yourselves, but that would take too long for our workshop today. So, Rafa, I see that we are recording now, so we're all set to go.
Yep, we're set to go.
Okay, so well, without further ado, today's agenda, I'll pull up here. Just 1 second. Oh, that was the slide I was supposed to push up earlier. Let you take a gander at that for just a second. Quickly gather in the when the workshop dates and locations are, but you'll be receiving invitations to those. And so here is our agenda. with the theme Detection, Response, and Management. And we're really excited to have a terrific lineup of 4 panels today. We feel that we have a lot to offer, and thanks to all of our speakers for sharing your time, expertise, and experiences. Each speaker has been given a time slot for their presentations, including questions, and we'll have a second panel then after that with a 10-minute bio break at 2:55. And so we'll really try to keep the things on schedule from here to go into the other 2 panels. So with that, I think we're ready to go into our first panel on environmental DNA. And so I'll turn it over to our first panelist, Courtney Larson from the US EPA lab in Duluth. Invite her to introduce herself and then discuss some basic questions about what eDNA can and does not do. We'll continue discussions with Don Eaton, Drew Dickert, and also with Justin Townsend. So Courtney, can you please take it away?
Thanks, Doug. Can you hear me all right?
Yes, loud and clear. Thank you.
Great. Well, thanks for having me. My name's Courtney Larson. I'm a biologist with the Environmental Protection Agency in Duluth, Minnesota. I've had this position for about 1.5 years now, and a main part of my research is how we can use eDNA in order to assess invasive species. And so I'm excited to talk to you about that today. I always, with this crowd, like to say a bit of my background was that my first job in science was as an intern with the Minnesota DNR doing watercraft inspections up in boat ramps up in Brainerd, Minnesota. And so that's what inspired my career going to grad school and coming back, and now excited to share this information with this crowd on eDNA. I was supposed to give a talk today, and it just, with some of the new rules we have with the federal government, didn't get it all improved in time. So I apologize for the inconvenience that I don't have slides today, unfortunately, but hopefully doing this new format of kind of a Q&A panel, going through some of the information more informally will still be adequate. I know it's not ideal, but it'll at least be informative. And so Doug and Russ, I'm not sure how, yeah.
Yeah, Doug, are you going to share the questions? And could you put your presentation into PowerPoint mode, into presenter mode, so that we can see them better?
Doug.
And then remember to check those that are online, if you're not muted, to mute yourself. We are hearing a little bit of background noise for some.
Yeah, so Courtney, while Doug is getting, well, I think he got them now. I was just going to start with the first question, but he got them out now.
Yeah, so my presentation was kind of supposed to be an overview of what eDNA is, what it isn't, how to sample for it and how you can use it. And so some of these questions will go through some of that information, just to start facilitating the discussion before I think we go into a larger panel and Q&A from all of you. So I think the first one is just how to know which species are good to target with eDNA as a tool. And so a lot of the invasive species that we're worried about in Minnesota, water bodies, have pretty good markers that are already published, and even have monitoring programs in the state already, but not everything has primers that are validated yet. And so if you're interested in using eDNA for a certain species, first figuring out whether there's a primer and a method that's already been validated is important. One really good place to look is the NAS database in USGS. They've actually started to accept eDNA detections in their database, and you can look and see if the species you're interested in has had reporting of detections on that, because you know that if that website has those detections, you know that it's a pretty well validated method. It's being used in monitoring programs. So that's a really good place to start, as well as just the primary literature to see whether there's qPCR primers. Another thing is just if there's a need. I don't think eDNA is the best tool for every single species, and it nearly never should be used alone. And so looking at your existing program and where are the gaps? Is it really hard to sample because they're really hard to trap? There's not very good gear available. Looking for species where there's a gap in your ability to sample them, maybe over certain spaces or certain times. Those are two good places to start, whether eDNA is right for you or not. All right, should I go on to the next questions, or is there any other panelists that wants to chime in with that one too?
Yeah, since I'm not hearing from the other panelists, I think we can go for the second one. So when, where, and how do I sample for eDNA?
So that's each system is going to be different. So this is where collaborating with someone who's already done eDNA work is really important. I think it's hard to go through eDNA alone, and so finding someone who can be a good partner and a collaborator is really useful. When to sample for eDNA? Some of the best times to sample for eDNA are when a species is spawning, because a lot of times the gametes are or larval stages. Those are all eDNA that can be captured, and so that tends to be some of the best times where you have the best chance of capturing them. Specifically, we do zebra mussel eDNA programs, and so we know that they release veligers in around August, and so that's been a great time to get really good eDNA signals, is when they release those gametes. Where to sample? There's two different ways to target where to sample eDNA for a certain species. One is obviously the habitat where you think you're going to get it, because eDNA is going to be released close to where that organism is living and moving around and shedding DNA. But also, with these aquatic water bodies, eDNA can pool in certain places just like any other particle. And so sometimes your highest eDNA detection is going to be in like behind dams or in eddies, or where there's areas where there's still water and where it can kind of collect behind a barrier. And so these eddies and dams have actually been really, really good areas to collect eDNA for a species, just even more so than where we think they may be living. So a lot of times when we sample for eDNA, we'll target both the specific habitat where we know we find them, but also behind a dam or behind kind of an outlet of a lake, or like an eddy if it's a river. How you sample for eDNA, it really varies by system, but most of the time it involves taking a water sample, something around 1 liter, and filtering it so that those particles, those particles are concentrated. And so it does involve, the actual collection itself is fairly easy and fairly similar to how you would collect any other water sample. The only added part is that there has to be some very serious decontamination and sterilization methods for your equipment, so that you know that you're not, adding DNA from other sources. So it's actually fairly easy to sample and filter for eDNA. Most people who have done aquatic sampling before find that it's pretty intuitive, but it does involve collaborating with someone who's done it already in order to make sure your methods make sense for the system you're in, what you're targeting, and what level of certainty and of detection you want.
Okay, thank you. And I think we're just gonna go with the last question, and then after that we'll go ahead and introduce the rest of the panelists, and they're welcome to chime in to any of the other ones. So the next question is, what steps do I need to take to know that an eDNA signal is real?
So with eDNA, you're not actually, you don't have a real organism in your hand. And so an eDNA signal does not necessarily mean that the species is there. And even if the species is there, that they're reproducing a stable, established population. And so one thing I really like to drive home with people who are just starting to get interested in eDNA and potentially implementing some eDNA in their monitoring is that you're detecting a strand of DNA that's a particular strand. You're not detecting something biological about a population. And so first, to know that when you do get a signal that says that that strand is there, that can mean many different things. It can mean, for instance, that there's a dead organism in there, because dead organisms that are an invasive species have DNA that are released just like a living one is. So there are multiple different ways that DNA could get in there, and what those kind of need to be teased apart using other methods. One of the ways to have more certainty that an eDNA signal is real, like represents a real living population, is following pretty robust decontamination and sterilization procedures. And so having clean gloves, clean equipment, and clean bleached, sterilized collection materials, so that you know that you're not getting any type of DNA contamination in any of your samples. One of the ways that you can validate that you're decontaminating and sterilizing enough is by running negative controls. And so you'll have blanks that are at multiple stages of your processing. So you have, we call them cooler controls, where we'll take sterile water and bring them out to the field with us, and then in our cooler, and then filter them just like they were a sample to see if there was any contamination just by the nature of being out in the field. We also run negative controls once we get back to the lab to make sure that our labs are fully cleaned and when we do DNA extractions. And so having lots of negative controls and replicates of the negative controls, you can have some level of certainty that there's not contamination happening. You also want to run positive controls to make sure that the signal occurs when you have the species there. So we will take tissue of the targeted invasive species and run analyses on that to make sure that there is a signal with the tissue itself. And so we'll also run some negative controls that are tissue of closely related species to make sure that those aren't showing a signal, even because they're closely related, even though they're not the same thing. So there's lots of things that you want to do along the way of your eDNA monitoring to be as sure as possible that the signal is representing something real and biological. Another thing is to do repeated sampling and using multiple gears. So for instance, we've had, with the research team I work with, we've had invasive signals of new detections that we weren't quite sure how to interpret, because it probably was just contamination, but we weren't quite sure. And so what we did is we go back and take samples again using a different molecular method. We also go and do physical trapping, and we also spread the word to local managers to see, hey, we saw this. Does this make sense to you? Is there something contaminating that you can think, or do you think this is real? And so I think that good communication among managers also helps to know whether it's real or not, and not putting alarm bells on when we just get an eDNA signal without a physical specimen, but get our kind of our just a little bit more sensitive to things. Like, yeah, maybe we should be doing a bit more trapping and communicating and figuring out whether it's here or not. It requires really good communication when you're dealing with an eDNA monitoring program.
I'm muted. Sorry about that. Well, thank you for that quick overview. I know that it's difficult without the slides, so I really appreciate your flexibility on that. I think at this point we're going to hold off on questions, and I just want to invite the rest of the panelists to join in. So if Drew, Justin, and Don, if you want to turn on your cameras and introduce yourselves at this time, that will be great.
Hi, folks. I guess I can start. I'm Don Eaton at Minnesota DNR, and I'm a colleague of Doug and Tina and Hoffa in the invasive species unit. I also work in the lake ecology unit, and I'll be talking a little bit about our response to signal crayfish, which is a new invasive in the state. But yeah, that's who I am.
All right, thank you, Don. And then Drew, I think you're getting ready there.
Yeah, I'll jump in next. Andrew Descartes, AIS coordinator for Carver County. Sorry, looks like my lighting is kind of bad today, but you can sort of see my face. And I guess I'll be talking a little bit about kind of how we use eDNA sampling and analysis here as part of our AIS kind of monitoring and early detection rapid response plan.
Did I go?
Yes, you may.
Sounds good. I'm Justin Townsend. I'm the Ramsey County AIS coordinator, and I've got Owen, our new AIS coordinator intern, who started all of 4 hours ago. So jumping in on the call as well. And yeah, working with Andrew and others, just trying to figure out how to get this eDNA thing into being a thing that we all use. So we can go ahead and talk whatever questions we got.
Okay, and I think, Doug, before we go, I think if we just get off of presenter mode so that everybody can see each other's faces for the panel, I think we got the questions right here, so I think we can just ask them. You think that's good with you? All right, sounds like that's good. Okay, so before we go on, I know that all of you work with different species. You all do different. You all use eDNA differently in your program. So I'm just wondering now, with any of the questions that Courtney just shared, do you all deal with different species? Is there like something different that you need to think about based on the species that you're working with? And this is just with the questions that she just answered. I just wanted to open that up for you all. Especially Don, I know that signal crayfish might be a little bit different compared to zebra mussels. So I'm just curious, yeah.
Well, sure. And I don't know, Courtney, if you have any recommendations in terms of time of year to sample. I know that luckily, we're not the geneticists here at the Minnesota DNR, but we have great partners at University of Minnesota. I imagine you also partner with Minnesota DNR as well, but they're the ones who actually do the genetic work. And we know from Gretchen Hansen's lab that, for example, for rusty crayfish, I think late summer is like peak eDNA time, or maybe the best time to monitor in general for them. But and in a little bit, I'll give a little background about the signal crayfish situation. But do you have any like general recommendations beyond, just having good collaborators that know when the best time to sample is?
I think that it really helps to have good collaborators who know the biology of the species along with the molecular side of things. In general, and it does take trial and error. I know Gretchen Hansen's, that was a whole study where they went every single, a few weeks out and found the best time that worked for each different species. In general, what I've seen is that for invertebrates, it does seem like August and September, late July is a really good time. But for fish, because spring tends to be the spawning season, if you can get it right around when that species is spawning, it tends to be like springtime is a better time potentially for fish eDNA. So I think having a discussion about the life cycle, about when you would expect, gametes to be released or when you'd expect there to be peak activity, it's different for everything.
Great, thank you. Yeah, and one thing I think I heard from one of our collaborators is that in terms of like, I'm going to talk about crayfish, I'm afraid, but when they're metabolizing, when they're eating a lot, when they go into mating season, which is sort of late summer, that may explain why there's so much DNA being shed during that time period. So maybe that's why that's a good time to look for crayfish if you don't know they're there, if that makes sense.
Okay. Yeah, thank you, Doug and Corny, for that. So I think we're gonna hear a little bit about our local, how eDNA is being used locally. So that's why we have Drew and Justin here. They both work with eDNA in their programs. So one of the questions that we have for you is how and when did your program start using eDNA for AIS work? And how do you see eDNA now? And the question is also in the chat if you want to also see it as well.
All right, well, I can take a stab at kind of kicking us off here. This is Drew in Carver County. So in Carver County, we use eDNA as part of our so it's not a large component of our AIS program, but it is a kind of key component of our early detection and rapid response strategies that we use within the AIS program. So it's kind of one tool in the toolbox when it comes to kind of surveillance and early detection. So we use it right now exclusively for zebra mussels. We haven't really expanded to other species. We may at some point. So kind of how we fit that in with our monitoring program, we sort of integrate eDNA collection with into our, like I said, our early detection program. So we have, our staff and our interns conduct all types of different surveys. Some of them early detection surveys, some of them are just macrophyte surveys. So we try to integrate the eDNA sampling anyway, while we're out doing other types of work. And then also kind of usually in that late July, August, kind of more productive time of the year. But you do have to be careful. You don't really want to go taking water samples in mid-August when you're dealing with like harmful algae blooms and such. So we've run into that issue a few times. But anyway, it's just kind of a way to enhance our early detection and surveillance, like I said, tool in the toolbox. And we can kind of complements the other traditional surveys, physical surveys, which we do as well. We'll go out to kind of priority lakes usually and do rake toss surveys for vegetation. But while we're out there, we'll jump in and do zebra mussel surveys, usually snorkel surveys, or just kind of jump down there and feel around with our hands if we can't see. And then with eDNA, we usually have a budget to do about 4, 5, 6 samples a year. It's not a ton, but we kind of, it depends year to year on exactly how we choose which lakes to get sampled. A lot of times it's, we'll think, okay, last year we had a positive eDNA on lake, let's just say Lotus Lake, for example. We'll want to come in this year and we'll want to check that again, because last year the eDNA Q was the only monitoring that came back positive. So we didn't find any physical specimens or velagers, but we had eDNA. So what exactly does that mean? We're going to want to come and test that again, along with the other, the traditional surveys. So it's a way to kind of confirm. You can't really confirm absence of the eDNA, but you can sort of confirm the presence of it. And again, I guess it doesn't really necessarily mean that there's zebra mussels in the water right when we took that sample. It means that there's eDNA there now, and there was likely the organism in that water at some time. So let's see, what else can I talk about here? We don't only use it for early detection. I've been kind of experimenting a little bit. I'll use Lotus Lake again for an example. It's a lake that has sort of 3 different lobes to it. They're all connected, but we sort of treat them as 3 different like areas of the lake, and we get different quantity of eDNA. I'm still trying to figure out how that works and how to compare that, but we can sort of track how much eDNA we're picking up from the sample sites year to year, and if that's changing, and if that means anything. Maybe we can understand if that means anything. And we bounce a lot of this stuff off of the director of the lab where we send our eDNA samples. And he's a guy down in Colorado, Pisces Labs, and we've been using his lab for quite a while. I think we started doing eDNA in 2017 or 18. So I guess that's kind of a general overview, and I'll maybe let somebody else pop in here.
Yeah, I think that's a good overview. I ditto everything that you said on what you're using it and why you're using it and the use cases for it. Maybe I'll just talk a little bit about where I'd like to see it go. I think it's definitely a powerful tool, and we've been weaving through in these different scenarios of people. We connected with Gretchen's lab and we're utilizing their study as our eDNA for a while at some sites. We were using Pisces. We were trying to work directly with the UMGC, the Genomic Center, to have it as a service for them, or to provide it, to have a local lab. And there's also one that the name escapes me. I'll try to find it here and throw it in the chat, but I think it's Equos down in Austin, Texas, that does it as a service as well. I don't know if anybody's used that in a while anyways, but I believe they provide it. I think the next steps in this is creating it as more of a service, because like Drew was saying, it's still just out of reach price wise for us to use it regularly. So it'd be really nice, speaking of timing wise, to be able to do it maybe a little bit earlier than what would be ideal, so that we can, if we do catch it and we do find something, say at a boat launch, can we do some treatment around it? Because right now, if we're doing it in July, that turnaround time might be several weeks. And then you're talking being end of August, beginning of September. And if you don't have your ducks in a row, you know that window closes really quick for treatment. So it's kind of then like, well, then why are we even doing this to begin with? So finding what we'd like to do is leverage our collectiveness in the near future, so that instead of sending individual samples in, or sending 4, 5, 6 samples in per county, can we fill a whole well and get some bulk pricing through whatever lab that happens to be? And can we do it multiple times a year if we get the price low enough, or find some price guarantees on getting this all together, so that we can utilize it more as a tool? Because agreed with what everybody says is, this isn't the be all end all, but if it's to be a tool that we use to get out there and get more eyeballs in the water, we have to do it a little bit earlier and more often than we're currently doing. Plus, looking to the future to like metabar coding and different things where we can do multiple species and shotgun this a little bit better. It's going to be super helpful for us if we're set up on the front end with this is how we submit these samples, and this is the price that it is, so that we're set up for the future, because it's coming at us really, really fast. This technology is changing extremely quickly.
Okay. Go ahead.
Oh, I was just going to respond to a few of those things because I think you're right on, Justin, like with where the field is going, absolutely, and like where the needs are. I think that echoes like everything that I've heard and experienced myself with this. Something to keep your eye on. So there actually was a national eDNA strategy that was published last fall or winter. It came out of the White House, and it was a multi-agency federal plan for basically answering some of those questions that you had of like, how can this actually be implemented on the ground? Part of that was federal agencies submitted a plan for implementation. That plan was submitted in November, and we are still waiting to hear what will happen with that implementation plan. Obviously, it's up to the new administration now. But what I'll say with that is the state of California did a very similar exercise. And actually, the state of California has an eDNA strategy where different agencies are actually collaborating together to develop a statewide, exactly what you're talking about, a strategy in order to use resources, have shared lab spaces, to have shared lab protocols, to standardize across different counties, different areas. I think that that's something that could be mirrored in Minnesota potentially, and that would be really probably a benefit to a lot of different groups if there's a way to like really strategize as a state.
Yeah, Courtney, that's great. Great news. And Justin and I have been kind of talking about this back and forth for a few months now. And when I attended the NASMA conference when was that, October or something, there were some really good talks on eDNA, and specifically a few were from some folks with USGS and Fish and Wildlife. And they did talk a little bit about the new the eDNR. Geez, I can't talk. DNA. I kept saying DNR. eDNA framework. So one of the things that are supposed to be coming out of this is sort of a cooperative lab research / potential place we can send samples. They talked about that being kind of one of their main aims, I guess, of opening this lab. So kind of what I've been doing is just waiting to hear what comes out of that framework. I'm sure everything is progressing quite slowly right now up at the top of the federal government. So yeah, I guess what I just chimed in to say I'm excited to see what's going to come out of that, and also in the lane that excited to see what's going to come out of more meta barcoding research. I think there's a really awesome future in it. I think that there's capability of learning so much about interactions between different species and their surroundings that you can get from kind of genetic analysis on an ecosystem level, which I just kind of want to nerd out about a little bit. I think it's great. So anyway, I'll shut up now. Thanks.
No, thank you. That's great to hear. Do you have something to add, Don? I see that you unmuted yourself.
I unmuted myself. So let me just backtrack a little bit, because our situation here at the invasive species unit is a little different. Like ideally we would also be doing regular monitoring, like you guys are trying to set up something like that. But a lot of times what happens, like in the case of, I'll just use the signal crayfish, but it's sort of a new invasive to a specific invasive species. And I don't know if you guys know, but it's native to the Pacific Northwest. The first record of it was in fall 2023 there near Alexandria in one particular lake. Just 10 individuals were found. But so we have this new invasive. It's a known, very dangerous invasive in terms of its impacts. It's all over Europe, was introduced. It's in Japan. So huge, very problematic. So in the invasive species unit, what we end up doing is we're responding to a specific new invasion. And so that's where the eDNA came in, and Gretchen Hansen in her lab immediately got funding to look at eDNA surveys. Unfortunately, it was late in the year, so we couldn't get any in the fall that it was discovered. But she was planning for which was summer 2024, to do the surveying. But so I wanted to mention that one of the first things that Eric Larson, who's our collaborator at University of Illinois, wanted to do was to get the specimens we did have and do mitochondrial DNA analyses. And the purpose of that was to sort of optimize the eDNA test or assay. Because there's numerous, like Courtney was saying, there are numerous tests depending on what species you're looking at. A lot of work's been done with signal crayfish elsewhere. And so he wanted to get, okay, what's the test we need based on the genetic strain we have here in Lake Winona there near Alexandria. And so they narrowed it down to this, to a test which had already been developed in California, where they've got both invasive signal crayfish and viral crayfish, which are native here but invasive out there. And they're common here in Minnesota. So Eric wanted to use a test which would be able to distinguish between signal crayfish and our native viral crayfish. And so there was that kind of optimization stage before doing the ED analyses. And then the other thing that's a little different, and so then like we went out and did a lot of trapping over the summer, trying to physically catch signal crayfish. And that was DNR, that was University of Minnesota and Douglas County. And after thousands of trap days, we caught no signal crayfish. And during the whole summer, Denver Link, who works in Gretchen Hansen's lab, had developed in previous years these little handy dandy kits for citizen scientists to collect samples, eDNA samples. So that was very convenient. And so all throughout the summer, folks from lake associations, the University of Minnesota folks, were collecting samples. I think it was about 140 samples collected from April through October. And those are when we just have some results now. And we've gotten 2 very weak detections of signal crayfish from Lake Winona, where the signal crayfish were originally found. And I'll see what Courtney thinks of this. But by week, and this is the geneticist telling me this, but they do several replicates, as you mentioned. Only one of the replicates in 2 samples had a detection, and it was very late in the run, which Courtney can explain this better, but it just means there's probably a low quantity of eDNA. And so one of the hypotheses, there's lots of explanations, but one of the bad news hypotheses is potentially what was detected are some juveniles which managed to survive over the summer or over winter and are possibly in the lake where they were introduced. And so, yeah, what's my point in all this? But so we're dealing more like kind of an emergency detection level as opposed to monitoring. And it's obviously been very useful. We did massive amounts of trapping, no adults, no signal crayfish captured, but now we've had this detection. So obviously it's important that we continue surveillance in Lake Winona. And we also were sampling in close by connected lakes or lakes just nearby as well. So all that was going on. So a slightly different strategy in response to a specific invasive, which there's a lot of concern about. I mean, there's concern about all invasives. I guess I don't want to rank them, but there was a lot of concern about this one just because it had such a huge impact in Europe and Japan and elsewhere. So yeah, I'm not sure what my final point is, but just want to point out like it's been very useful in kind of this sort of emergency detection sort of situation as well, because now we know we need, we're not out of the woods. At least, we could be, but we don't know for sure.
Yeah, I think, Don, you're speaking to something that was a surprise to me when I first started with eDNA and qPCR, and that it's not black and white. You don't get just like, yes, it's in there, or no, it's not. Like, it's like, well, it showed up on one of 8 replicates, or, oh, it showed up really late. So is that real or not? And it's way more murky than you would expect for something like as something that's so molecular. And so I think exactly what you're saying, like just keeping sampling and continuing to monitor and having those, the alarm bells are sort of ringing, but they're not terrible yet, kind of a thing when it's in that gray area.
For sure. And just so you know, of course, just as you mentioned, Courtney, they were doing blanks and none of the blanks had any contamination. So there's always the idea, oh, well, this was just some contamination, but at least based on their blank samples, no contamination came back from the citizen scientists sending in samples. From the ones that I sent in, that could happen. I mean, we all used a specific protocol that Denver and Gretchen set up. We use gloves and like you mentioned, have a set procedure. So we're all sampling in the same way. But that was one of the alternatives which was mentioned. It could be a blank contamination, or it could be a contamination of some kind. They could be detecting on different non-target species besides the viral crayfish. Like calico crayfish also occur in this lake. They're pretty rare. But so one of the things that Eric Larson, the geneticist, is wondering about, okay, is this possibly a detection of calico crayfish? And he's now going back and I think retesting to see if that's a possibility. But there's so many of these alternatives or alternative explanations, which I guess is why, and one of your questions, Rafa, is why we can't just stop sampling or stop looking for these guys until we have, if we ever get a definitive evidence one way or the other. But we need to probably get out there again, keep trapping, keep looking.
Yeah, and so yeah, you're right. You're right, Don. And I know that we could probably keep talking about eDNA for a whole workshop, but I see that Doug's getting back on camera, so just to keeping track of time, right, Doug?
Yes, I am. Do Drew or Justin, do you have any final thoughts you'd like to add?
One really quick thing, Don, I really appreciate that comment because that setup is what is really going to help us. If we can rely on the feds and the state to do that research part of it and make sure that this stuff is available so that we can do the ongoing monitoring at the local level, I think that's a great a way to set it up that we can continue under that assumption for the near future anyways.
Right.
Yeah, and just to really briefly add on to that, I think that, Justin and I are natural resource managers, right? So we're not involved all that much in the research aspect of things. So for us as managers, having the state, the country, whatever, come up with more of a standardized protocol for using eDNA kind of based on what you want out of it. That's kind of what we're sitting around waiting for.
Well, and Courtney, can you give us like an idea of like what's the cost of the analyses and so forth? Just that's probably tough to say, but
Yeah.
like
Don,
what are we looking at?
yeah, Don. Unfortunately, we're right on time, and I know
Oh,
that
I'm
we could just
sorry.
keep going and going and going. But no, thank you for all of you for being part of the panel, and you all can definitely just keep talking in the chat. If you all can just hang around, and I'm sure that the attendees have lots of questions. So if you all have any questions, please send them out on the chat, and I'm sure that our panelists will be able to answer that. But we do have to keep going to our next presenters. And Doug, who do we have next? And again, thank you so much for being part of this panel.
Yeah, thank you all to all of our panelists. That was excellent discussion. Hopefully you can see my screen. So we are just a couple of minutes over, not too bad. But our second panelist is basically a discussion on invasive aquatic plant management. We'll have an overview of IAMP from Wendy. We'll have some case studies with Chris Jurek. We'll have some chemicals and some projects, discussion with April Londo, and finally, DNR detection and response plan by Tina Fitzgerald. So, without further ado, Wendy, you want to kick it off? I'll stop sharing my screen.
Okay, there we go.
There you go.
I will start sharing my screen, see if you can see it. Can you hear me?
Yes, we can hear you. Thank you.
All right. Can you see my screen? Should have my
Just came through.
presentation.
Yep.
Yes?
It is. Looking good. Go for it.
Excellent. Well, let me just get this up. Yep. Well, thanks everybody. My name is Wendy Crowell. I'm the Aquatic Invasive Species Management Consultant with the DNR's Invasive Species Program. I've been working with the Invasive Species Program for the past 31 years. About half of that time was as an invasive species specialist. And I'm going to talk about aquatic invasive species plant management. So the DNR is committed to working with partners to achieve these three goals. And we can use management to help achieve these goals. So that's prevent the spread, appropriate response with early detection, and then reducing the negative ecological, recreational, and economic impacts of AIS. So we do this. So management encompasses providing technical assistance, providing grants to help control, permitting chemical and mechanical treatments through the MPAR system, and supporting research that leads to improved AIS control techniques. And of course, we also do monitor and initiate some control projects ourselves, again, to support those three goals. So overall, the DNR Invasive Species Program uses this basic approach to invasive aquatic plant management. So invasive aquatic plants can cause harm to natural aquatic ecosystems, and they can also interfere with the use of those natural resources. And the management of invasive aquatic plants can be a good way to mitigate those problems, but it's also important to remember that the management has to be done in a way that protects natural resources and their use in the state. And so new methods and new ways of using old methods and products are continually being developed and evaluated to better achieve those goals. And then that's part of what we do as a program. So in order to protect lake ecosystems, the destruction, harvesting, or replanting of aquatic plants, and that's invasive or native aquatic plants, is regulated under Minnesota law. And there are two Minnesota laws that govern the destruction of aquatic plants. 103G.615, that's a statute, and then the rules, which is 6280. So 103G.615 defines an invasive aquatic plant management permit. And those permits are issued for the selective control of invasive aquatic plants to cause a significant reduction in the abundance of the invasive aquatic plant. Now, this is really different from aquatic plant management permits, which are issued for the non-selective control of any aquatic plant, which could be an invasive or a native. And that's issued for almost always for recreational access. The statute that defines these two kinds of permits also says that the criteria for how we issue those permits is laid out in Minnesota Rules 6280. And so Minnesota Rule 6280, again, it applies to both invasive and native aquatic plants. And when the law makes a distinction between chemical control with aquatic herbicides and mechanical control, it distinguishes between emergent, floating leaf, and submerged plants. So emergents would be the ones growing out of the water, floating leaf are your lily pads, and submerged plants are the ones under the water. And it also distinguishes between invasive and native aquatic plants. In general, chemical treatment requires a permit. Anytime you put a chemical in the water, if it's a public water, it will need a permit. The lake-wide limit for chemical control is a lot less than the lake-wide limit for mechanical control. So mechanical control, you can do small areas without a permit at all, 2,500 square feet generally. And the lake-wide limit is 50% of the littoral zone, while the lake-wide limit for chemical control is only 15%. A variance of, and these are just some of the limits and rules that are laid out, but they're some of the most important ones. Now, you can get a variance to these limits under special circumstances, and the control of invasive aquatic plants is one reason that can be considered for issuing a variance. In addition to those laws that govern the destruction of aquatic plants, there's laws that govern the use of pesticides for invasive aquatic plant management. And these are the general laws that govern the use of pesticides. The pesticide has to be registered by the U.S. Environmental Protection Agency. And remember, again, when I say pesticide, that includes an herbicide. Herbicides are just a class of pesticides. So it has to be registered by the EPA. And it also has to be registered by individual states. So that would be the Minnesota Department of Agriculture. It has to be used in accordance to its labeling. And then that use includes handling, mixing, storage, loading, transportation, as well as use, the rate that you put it on. And then that label is the law. And just to try to explain this, like if you have a shampoo and it tells you, it gives you instructions on how to use that shampoo. But if you don't use the shampoo the way they say, you're not breaking the law. But if you don't use a pesticide exactly how it's described on its label, you're actually breaking a federal law, and the penalties for doing that are quite severe. You know, $10,000 up to. So those are the laws. So now I'm just going to talk about the three types of management and compare and contrast them a little bit. So the three main types of management are mechanical, chemical, and biological. Chemical is the most commonly used method, followed by physical control methods, and then biological control is mostly for invasive aquatic plants. That would be mostly used on medium to large stands of purple loosestrife, though there are some efforts still to use a milfoil weevil for control of Eurasian water milfoil. So when you're thinking about what management method you want to use, so a management method should be chosen to achieve your desired outcome. So the first thing you want to do is think about what your goals are for invasive aquatic plant management. And then once you understand what your goals are, then you think about the species you have and the methods that are available to achieve those goals. So potential goals might be lake access or recreation, or maybe prevention of spread. And then there's also long-term control. That is one of the goals where invasive species are controlled and to provide ecological benefits, or sort of supporting native plant communities. So different control methods can be very effective at achieving these goals, but the effectiveness is really going to depend on the target plant, the size and location of the infestation, and the timing of the control. Like when you're doing the control will determine how effective it is. And of course, remember, we want to do the treatments have to be selective. So it's, you're going to have to look at what non-target species occur in the area. And then finally, of course, what resources you have available to pay for these efforts. When you're talking about pesticides or herbicide control of invasive aquatic plants, some herbicides are really effective at some plants. Milfoil is a dicot, so all the herbicides that are selected for dicots are effective on that. So then that affects your species, affects what chemicals you can use, and also maybe what timing. Curly pondweed has to be done early in the season if you want to prevent turion production, for instance. And then, of course, rate of application. With a lot of the herbicides we use, we found that the lower the application rate will provide more selectivity. So generally, pros and cons of chemical treatment. You can achieve long-term control, like with milfoil, which is a perennial. You can actually, there are some herbicides will actually destroy the roots, which is good. So you get more than one season of control. And it can be selective. Some herbicides, particularly at low rates, can be really selective. Cons, one of the most significant cons of herbicides that I think people don't think about enough is that an herbicide treatment can completely fail, right? Because the herbicide effectiveness, particularly at these low rates that we're really pushing that low rate, you have to have a suitable contact time. And if you have a small area that you're treating where there's wind or water movement, that's going to move that herbicide off before there's an adequate contact, you could do an entire treatment and really not get essentially any control at all. And then the sort of the flip side of that is that if you put an herbicide down and maybe it doesn't mix thoroughly, or it's gone in at a slightly higher rate, or you thought it was going to be selective at one rate and it's not as selective as you thought, then you can get non-selective control. So that's pros and cons of chemical. Mechanical control, and these are the two main mechanical controls. The harvesters, the big machines that mow the top 6 feet off, or, hand pulling with scuba divers or snorkelers. There's really different kinds of control, but they're all lumped together as mechanical control. And so what you can achieve is really going to depend on what kind of equipment you're using and what kind of area. Like, are you going to do a really large area, or do you just have a few plants that you'd like to pull out by the roots with scuba divers? I mean, that's something we use for star stonewort, and it can be really effective, particularly if there are only a few plants growing. And then so scuba diving is a good method to use for early detection, rapid response. But this giant, big mechanical harvesters are really good if what you want to do is control the plants and make sure it's not interfering with the use of the lake. Because remember, you can get a permit to treat with mechanical up to 50% of the littoral zone, which is different than chemical treatments. So pros and cons of mechanical control. So the biggest pro is that the results are immediate. You run your harvester through, or you send your scuba divers out, and those plants are removed. And the plants are removed. In fact, they have to be removed. If you do any kind of mechanical control, the plants have to be removed. Even if you're doing the kind without a permit, you can't just kind of cut them off and let them float around. And then it's not like chemical, you don't get a failure. But of course, the con is that the plants will grow back, particularly if you're using a harvesting machine. Unless it's curly leaf where it's naturally going to die back anywhere. If you're running a harvester through Eurasian water milfoil, those plants are going to grow back in a few months. The harvesting is not selective. It's going to take all of the plants that are there. But again, it's, those plants are going to grow back. The scuba diving can be extremely selective, but it's very expensive and it can only really be done on small areas. Purple loosestrife. So I'm going to talk about, this is purple loosestrife, and those are purple loosestrife beetles there. I'm going to talk about biological control because this has been so successful. So the control of purple loosestrife has really been a successful example of a classical biological control. So as it says on the slide, classical biological control is the use of a non-native natural enemy to reduce the population of a non-native pest. And so, I mean, as you say, we're bringing yet another non-native organism to and releasing it. And so the use of biological control requires extensive testing. So that's testing in the native range of the plant and the biological control, and then testing under quarantine conditions in our country and testing against other plants to make sure that the organism is selective. It's completely selective. It's not going to damage the ecosystem at all. And then for purple loosestrife, that whole process took about 10 years to complete before it was deemed safe enough to release the insects into the wild. And even after they were released, the impacts of the beetles on purple loosestrife and native plants were monitored using permanent sites for several years. So the pros and cons of biological control are, well, if you find something, then it is selective, and it's also self-perpetuating and self-dispersing. So this is, once you get it going, it just goes. So it's very cost effective. But of course, that's after you have it. So now that you know purple loosestrife, I always recommend for moderate to large stands of purple loosestrife to use the beetles because it is really the best option out there. But the cons, if you don't have one already developed, is it takes a really long time to develop it. And the other thing is that you can do all that work and take a really long time and then end up without a biological control organism. So you can, the reason it goes through all that testing is to make sure that it's selective. But of course, some organisms that you think might be selective turn out not to be selective, or they turn out not to do very much damage to the plant. So then they're not a good biological control. And of course, it's not completely risk-free. There could be unanticipated host switching, though we have not seen that with biological control of purple loosestrife, even though it's been going on for decades now in the United States. So the best management option is going to, as I said, just depend on a lot of different factors. And new information about the plants, their biology, different management techniques are being researched and tested all the time. Companies are working hard to develop new tools for us to use and try. And we actively work on testing new products and new ways of using old products or methods, and also monitoring the results. Like I said, if you're doing a large treatment, you'll have to use a lake vegetation management plan or get a variance, and then that requires monitoring and testing. And so that's some of the testing that we do. So now, just at the end here, I'm just going to say, finally, if you've decided you have an invasive aquatic plant project you want to pursue, how do you get permission? How do you get an invasive aquatic plant management permit. So almost all AIS control projects are going to require an invasive permit, an invasive aquatic plant management permit from the DNR. So I'm just going to walk you through the steps. So the first thing you're going to want to do is determine exactly what your goals are, what lake you have, what control method you'd like to use, and what species you're going after. And then you got to get a map of the areas, really the areas we're going to treat, and preferably a GIS file showing those areas you'd like to treat. And so private consultants, commercial pesticide applicators, and in some cases, AIPM specialists can help provide these, and then decide if you're going to hire the work done, get a contractor, or try to do it yourself. I'd recommend a contractor, but some really small things can be done by yourself. And then if you already know that you'd like to do more than the limits outlined in Minnesota rule, for instance, if you want to do a lake-wide treatment with fluoridone to control Eurasia water milfoil, you're going to want to contact your invasive species specialist to discuss your project ahead of time. They may ask you, they're going to ask you for more information, and they're going to require some data up front so they can review the possible project. And that's, you're going to want to give yourself quite a lot of lead time to work through that process. But if you have all that stuff ready to go, the next thing to do is go to MPARS, the Minnesota Online Portal for Permit Applications. And this is the easiest way to go to MPARS. Go to the DNR website. And this is the DNR website. In the corner, there is corner over here, search bar, type in IAPM. And what you'll get is the invasive aquatic plant management permit web page, and it'll look like this. It's got a lot of information about IAPM permits, and it also in the bottom corner here has a link for the MPARS system. And that's, you can just follow the instructions through MPARS and you can get your permit. If you have any questions, what you gonna want to do is go to the DNR contact page. You can contact your local regional invasive species specialist. You can contact me, my contact, all of the DNR staff with the invasive species program. All of our contact information is on that, and it's on the DNR website. And with that, I think I have run over my time. So
Just by a couple of minutes.
yep. So
Thank you so much,
go.
Wendy. That was a great overview.
Oh yeah, you're welcome. Oh, I didn't have my camera on. Oh well. You didn't see me moving my hands.
Well, thanks again, Wendy. Really
yeah,
appreciate
sure.
it.
Yeah, you bet.
All right. Well, let's move on here. I'm going to just show the screen one more time here. And we'll now move on to Chris Jurek, who's going to talk about case studies of starry stonewort hand pulling. Take it away.
All right, I'm just going to set up my screen really quick here.
Okay. Looking good.
Okay, you can hear me too?
Can hear you too. Take it away.
Okay, great. So I'm Chris Jurek, invasive species specialist. I'm out of St. Cloud. I've been going, this is my 13th year working in this same position. Today's presentation, I'm going to talk about starry stonewort detection and management in Minnesota, but I do have some slides on hand pulling starry stonewort. I always like to start off my presentations of what is starry stonewort. Maybe some of you are super familiar with this plant. Some of you may have never seen it. It's really unique macroalgae. It's not my favorite plant, but it keeps me busy. It's a green macroalgae. It's found in the plant kingdom. What makes this different, it's non-vascular, but it's plant-like in function and growth. Some things to look for are whorls of branchlets. It has no stiploids, cortification, or spine cells in this genus. It's similar to your native stoneworts in Minnesota. So when I say it has none of those properties, not properties maybe isn't the right word, but it's not your muskgrass or charophytes that are native to Minnesota. And I have a picture there of Cara, which kind of has those bumpy structures that you can see underneath a microscope. And you do not see these when you look at starry stonewort under a microscope. Some unique things to look for when you identify starry stonewort is these uneven brack. On the bottom right of my screen, you look for these branchlets and you look for that uneven forking appearance is what I look for when I try and identify starry stonewort. It doesn't always have those bulbils that we always hear about. It also has long internodes, which will probably show up here better. So an internode is between the nodes. It's quite longer than your native stoneworts in Minnesota. A lot of times you find native stoneworts with a starry stonewort, so it's sometimes hard to tease the two apart, especially when you have native stoneworts and muskgrass all growing in the same area. It does have a smooth stem, which is something to look forward to. And when you pull the starry stonewort out of the water, it's pretty erect. It's pretty firm. It kind of holds its shape. Let's see, what else should we talk about? This is my favorite illustration. I think it's from Golden Sands out of Wisconsin. I should have edited that to show that reference there. Reproduction and dispersal. Fragmentation is a huge way of spread of this invasive species. It's moved by boats, trailers, anchors, docks, lifts, and gear. Most of the time we find new infestation at boat accesses right beyond the cement slabs. It's like the boat drops in and boom, it drops off the trailer. That's where I find it probably 90% of the time. It has these specialized structures called bulbils. They're white, star-shaped, approximately a centimeter. They're located on the threads at the base of plant, found at or below the sediment surface. So there's a picture on the bottom right. But there's also these green bulbils. They're found at the main axes and branchless nodes, where in my office, I was trying to grow one out and see how long it took to grow a new starry stonewort plant. It didn't take very long. The phenology and growth patterns. When this was first detected in Cronus in 2015, so 10 years ago, me and my technician went out and drilled some holes under the ice, and lo and behold, we found tons of starry stonewort, green starry stonewort, growing under the ice with bulbils. So we discovered it's in a lake all the time. Bulbils found at any time, but higher bulbils are found in the fall when the biomass is at its greatest. These are just some observations on Cronus, where I spend most of my time learning more about this plant and doing monitoring. It does grow in water up to 9 feet, but it seems to mat at 7 feet. Cronus is more of your mesotrophic lake, so that could play a part into some of these observations, and it might grow a lot different in a different water body, especially maybe up north where the lakes are more oligotrophic, or might grow differently in southern Minnesota. So I've been monitoring Cronus for the last 10 years. I haven't updated this slide for a while, but it has spread from the location of infestation on the east side by the boat access westward, and currently it went from 15% to close to 80% of the lake has starry stonewort. I should say that's the percent frequency found in the lake based on point intercept surveys that we've done. Just like any other invasive species, it does have some impacts to recreation, swimming and boating especially. This is really thick. It can mat both vertically and horizontally in a lake, which makes it very challenging to boat and swim through. A lot of people ask, what does the DNR do when we find a new infestation? Just like any other infestation, we respond to the report, we go to the site, identify the species, confirm its presence. Then we start working with our partners and stakeholders and post signage at the boat access. Typically, we work with lake groups and coordinate funding, or there may be grant money to conduct management or monitoring of the species. There's quite a few lakes that there's no management occurring. From then, the contractors are hired to apply pesticide or hand pull the starry stoner at the sites, and we continue to work with our local partners and contractors to control the starry stoner. We encourage annual monitoring just to determine the success of the projects, of the management projects. Most infestations are found at boat accesses, so it's really an important site to monitor just because the boat traffic in and out of those areas. You want to make sure that we can put down pesticides to reduce the biomass to prevent and limit the spread in and out of the lake. Monitoring new infestations. So once we determine its presence in the lake, we kind of go another step. Depending on the situation, typically we'll go out and do a quick, I shouldn't say quick, it takes sometimes a few days, a meandering survey of the shoreline, littoral area of the lake. We can do point intercept surveys, but it's also harder to find sparse populations using that type of method. Actually, the best way to find new infestations is snorkeling and scuba diving, especially near public water accesses. That's where, like I said, majority of our infestations are found. To date in Minnesota, we tried quite a few different management techniques, including suction dredging, dash, copper hand pulling projects, hand removal, mechanical harvesting, and then no management. To date, the most popular methods are copper and hand pulling. I'll get into it a little bit later. Both all methods can work, but some are a little bit more cost effective. In terms of managing, there are quite a few things to consider, and this is kind of the same for any invasive species. Size of infestation. If it's a large infestation, I would say more than a couple acres, you probably won't want to hand pull. Personally, I've hand pulled starry stoner and I wouldn't want to go more than an acre. And it also depends on the density of the starry stoner. And if it's intermixed with natives are some considerations. You also want to consider your native plant communities. If there's a lot of muskgrass and native stoneworts, sometimes those pesticide applications can nuke those areas and allow for starry stoner to come in and more aggressively evade those areas. So you have to kind of balance if it's doing more harm than good. Timing, starry stoner treatments. Most of my lake groups, I tell them to start at least looking in July. Pesticide treatments and hand pulling, start them in July and run through September, and do this monthly. Management is monthly. At least that's where we're seeing the best success, is going in every 4 weeks. It's kind of like cutting grass. It just comes back. You can only get annual control using any of these methods. Which is really unfortunate. You don't get any carryover impacts with pesticides or hand pulling. Evaluating management is kind of similar to doing our pre-survey assessments. We can do point intercept surveys. We've done copper monitoring. We want to make sure our concentration exposure times are meeting the label rates. Usually they're 1 part per million with copper is what we run those treatments at. For small infestations, snorkeling and scuba diving, we can go to the axis and look at presence, absence, height, percent cover of starry stoner, just to see if we're getting any reductions in biomass and percent cover of starry stoner in these areas. We did some sediment sampling for bulbils and macroinvertebrates on Lake Cronus. That was a 2-year study. I won't go into detail there, but bulbils are very variable in the sediment. They're not the same as turions. So with endothel treatments, you can see good reductions in turions over time. Bulbils, I think that they're more variable. And we also did a lot of biomass measurements, what I'll get to a little later. And we kind of collect all this data to evaluate trends over time and to guide future management decisions.
Hello, Chris.
Yep.
Yep, we're over a couple of minutes here, so if you could.
Oh, shoot.
wrap up.
Okay.
Thank you.
I'll just do one more slide. So this is my hand pulling slide. So our most successful projects were hand pulling. So on Grand, Westlake, Sylvia, Pleasant, Carnelian Lake, and Rice. Here's a picture before and after. So very selective way of management. And I'm just going to show you my last slide here. This shows biomass reductions using hand pulling. So usually the first hand pull in July pulls most of the biomass out of the lake, and then the divers can go in on a monthly basis and just hand pull. And it's been very effective. So if anybody has any more questions, feel free to call or email me, and I can talk about this all day long. So thanks for listening. Sorry to cut this short.
Thanks, Chris. Great presentation.
Thanks.
Obviously, management of starry stonewort is very tricky.
Yes.
All right. So again, I'll just share my screen real quick here, and we'll go on to our next presenter, who is April Londo, who's going to talk about some of the chemical herbicide projects she's been working on. Thanks. Take it away, April.
Helps to unmute myself.
We are seeing your notes. There we go.
Got it?
Looking good. Can hear you.
Okay. Can you see my full slide?
Nope, we're
Or what are you
seeing
seeing?
notes now. We were seeing it all good just a moment ago, but now we're seeing your notes. Now we're seeing it good.
Yeah. Hold on. Get out for a second.
I've had the
This
same
work-
problem.
This worked just the other week, so I don't understand what's happening.
It's
April,
team.
open up your display settings and there should be an option for switching the screens. It's at the top left.
Left? This one?
So when you had your screen previously with all the black in your notes.
Mhm.
And now
Oh
we
my
can
God.
see your whole slide. So
Jeez Louise. This has never happened to me before. This is frustrating. Okay.
This just happened to me
Up
last
here?
week, April. You got my
yeah, so
Display
on the
settings.
left side, your display settings, if you can swap the presenter view and slideshow, that should work, but I don't know why it's grayed out.
Okay. Well, we're just gonna roll with it, I guess. And
There you go.
sorry guys, I'm not helping the time crunch here. Otherwise, I'll just have to wing it without my notes, and we'll see how much I remember.
Do you have two screens?
I don't. I'm at home.
Hey, April, can
Mhm.
you present in Teams? Sometimes when you present in Teams, it shows everyone the presentation and you just see your notes.
What do you mean present in Teams?
Like if you go to the PowerPoint up in like the upper right-hand corner, you have an option to present in Teams.
Let's see.
Should be up by like record, share. No, like up.
Yeah, hold on one second. I just want to see if this is going to work. But it didn't. Okay, Angelique, I'm there. What?
There's a presenting
Present
teams
in
on the
teams?
left, up left.
Yep.
Or
Yep.
there's
Okay.
The one that you had your
I'm
mouse
able
by.
to present. Okay.
That looks good.
Except it's in presenter mode.
It's not for
Isn't it?
us.
Oh, it's not.
Yeah, we see the
Okay.
presentation.
Beautiful. I'm sorry about that, guys.
No
Well,
No,
worries.
I learned a new thing. That's good.
Jeez, I honestly just presented last week and it worked flawlessly. So I don't know. But anyways, my name is April Londo. I'm the invasive species specialist covering the counties in and around the Twin Cities Metro. Thanks for asking me to speak today. This is going to be a whirlwind for you because I'm talking about 3 different products. And in the interest of time, I'm only going to be talking about one case study for each of these. So fluoridone and fluoropyroxafen benzyl are both going to have your Asian water milfoil as the contact or the target plant. And then panoxylum, also known as galleon, is going to be targeting curly-leaf pondweed. So Wendy did a great job at introducing our program. And just as a reminder, our main goal is to minimize the harmful effects caused by invasive plants while protecting natural resources and their use, right? So we implement adaptive management, and this includes continuing to study about the biology of these species, monitoring new and existing projects, trying to understand current methods that we are using, and working with researchers to test new products, new use patterns for existing products, and then adapting as we go. So you observed some of Chris's maps, right? So I just wanted to go through the standard method to track aquatic plant abundance and frequency over time is this point intercept survey. So you use a designated grid to collect plant data, including rake abundance, and then all species observed. And you're using a rake rating, or at least at the DNR, we use a 0 to 3 rake rating, as you can see on the upper left right there. And then this will calculate the abundance, and the dot densities are referring to how much of that plant you're seeing at each location. So these will come in handy later on in my talk as we go through. Fluoridone and I were both established in 1986, and it's applied at a bay-wide or whole lake level because it's of its ability to disperse and move relatively fast in the water. Plants need to be exposed to the product for upwards of 90 days for milfoil control. And then we also use it for curly leaf control, and the plant has to be exposed to it for about 30 days. This product is something called a systemic herbicide. So it's translocating to the roots and it's killing the entire plant. This product has also been used to treat hydrilla, water hyacinth, and then, like I said, we use it to treat milfoil and curly leaf pondweed. Maximum label rate is 150 parts per billion, but we definitely do not use it at that. The DNR started using fluoridone over 30 years ago. We can thank Wendy for a lot of this spearheading of adaptive management. This is the poster child of adaptive management in the state. Shortly after fluoridone was registered with the EPA, the Minnesota DNR started a full evaluation of the product, which included research reviews, public meetings, and developing a research design to evaluate the product in mesocosms and then also in lake studies. They spent 4 years studying different concentrations between 10 and 20 parts per billion in 13 different lakes. What they gleaned from these studies was pretty bleak for native plants. Although there were some resilient plants that survived at higher levels of fluoridone, like Robbins pondweed, naiad, water stargrass, and leafy pondweed. They observed decreases in milfoils, elodea, large-leaf pondweed, flat stem pondweed, and coontail. So they knew that they needed to make some changes here. So obviously using the product at 10 to 20 parts per billion was not feasible. We want to be selective in our control. So in 1997, research was being conducted in Michigan, and they determined that milfoil could be controlled as low as 5 to 7. So that was kind of their lower threshold that they were using. Shortly after, the DNR wanted to implement this and did 6 parts per billion in 3 metro lakes. But we wanted to know if we could go lower and still see control. Initially, there was a major pushback to go much lower than 6 parts per billion because people were concerned that they weren't actually going to be controlling milfoil. But the DNR and others kept on trailing ahead. And in 2013, Minnesota DNR developed a guidance for fluoridone between 4 and 4.5 parts per billion. But in 2016, we tried fluoridone between 2 to 4 parts per billion for the first time in Minnesota, which was on Crooked Lake, and this will be the focus for the case study for fluoridone. This is a slightly eutrophic metro inland lake where EWM was first detected only 3 years after it was first detected in Minnesota. In 2016, Eurasian water milfoil was hovering just below 60% in the lake. So that's frequency of occurrence. So of all the points that we went to, 60% of them, we observed milfoil. Target concentrations, like I mentioned in this lake, was 2 to 4 parts per billion. These are really good representations of what the plants look like before and after. So on the left, you can see they're very healthy. There's a lot of green growth, new shoots coming off. And then post treatment, you observe kind of this dark decaying matter. It was starting to defoliate. And then here's a good representation of, what we use to get these plant data, which is a double tine rake. Super innovative. Here's a graph completed by Coon Creek Watershed District. The treatment was done where that dashed red line is, right? And as you can see, 6 years after the treatment was holding under 5% frequency of occurrence for milfoil. Here is a table showing you all the different plants that we observed. There were actually 15 taxa observed from 2016 to 2022, but we just included ones that were over 5% frequency of occurrence. So you can see that most of the native plants stayed stable. The next product I'm going to talk about is fluoropyroxafen benzyl, or Priscillacor. So this is the only product name right now. It's not off of patent, so it's called Priscillacor right now. This product, like for fluoridone, is a systemic herbicide and will kill the entire plant. And what it does is it mimics the activity of the plant hormone auxin in a new way, unlike 2,4-D and other auxin mimic herbicides. It's susceptible, like the susceptible plants will show a mixture of atypical growth. So it might have larger or twisted leaves. The stems will be elongated, and the fragility of the leaf and shoot tissue will be evident. Our case study here is Lake Jane. This is a small, deep lake, mesotrophic lake. It's 14.2 kilometers northeast of St. Paul. So in this lake, we did 2 different treatment areas that I'll show you in a second. And milfoil was first reported in 2012. It was suspected to be hybrid. And later we did observe through Newman's lab and Thune's lab that we did have hybrid water milfoil, and that was confirmed in 2020. And we had nuisance conditions in 2018. So what we did is we set up 2 different study plots, and they were actually treated in 2 different years as well. But pre-treatment, the hybrid water milfoil in the north plot was 72%. In the southern plot, it was 58%. So after treatment, 1 year after treatment, we observed in that northern plot a reduction to 1% and 8% in that southern plot. Plants were devoid of leaves, so you can see in that picture on the bottom right, it's just like a goop of just black stems, possessed dark roots, like I said, and were presumed non-viable. Negative impacts that we did observe, there were some non-target impacts, and Wisconsin DNR and literature also alluded to this. So floating leaf pond weeds are susceptible to Priscillochlor, so keep that in mind if you're going to be using it. And our positive species that actually did better 1 year post-treatment were the 6 listed here. This was published in 2022, so if you would like to read the full study, you can search Couture et al. 2022, or you can search the actual publication title. For gallium or panoxylum, so this is another pesticide that we're just going to use gallium right now. It's interchangeable with panoxylum, just for in terms of this presentation here. But it was first registered for terrestrial use in rice fields and turf in 2004. And in 2007, it was registered for aquatic use, but was primarily used for hydrilla, water hyacinth, and milfoils. To date, only a few published laboratory studies exist. And these studies were conducted in between 2011 and 2013. And they found that varied exposure times and concentrations could impact Illinois and Richardson's pondweed, but water stargrass may flourish after treatments. And it was also successful at controlling curlyleaf pondweed, even though it wasn't initially used for that target. We had a few case studies. So if you want to know more information on more of these case studies, I can share them with you. Just reach out to me. But this is Medicine Lake. It's a 924.37 acre lake. 43% of it being utilized for aquatic plant growth. It had its maximum depth of 49 feet. And the target concentration for panoxylum in this lake was 17 parts per billion. So this is a table showing pre and post what happened in this treatment plot. And let me just back up. The treatment plot is where that star is, where the dashed lines are on the lower right-hand side in Medicine Lake, just to give you some context. So over here on the left-hand side, we have our taxonomic names, right, our common names. And we went out 4 different times, pre and post for the first year, and then pre and post for the second year. And the things in parentheses are showing significant increases or decreases in the frequency of occurrence of these plants. And the red lines are where we treated. So significant difference were noted. And as you can see, we had decreases in curlyleaf pondweed, Eurasian water milfoil, and sago pondweed. And then we had a significant increase in muskgrass, elodea, and wild celery. This is a good visual representation of what happened in 2022 and then in 2023 when we went back out there after 2 treatment time periods. So over on the left, you have in 2022, pre-treatment is up on the top. So you see a lot of the yellow dots are where we observed curly leaf pondweed. After the first treatment is on the bottom left. And then over on the right-hand side is after that, after treatment in 2023. So as you can see, there aren't many dots, and where you do observe them, we found sparse curly leaf growth. There was an 85% reduction in curly leaf pondweed frequency. So this is a graph just showing how abundance changed. And as you can see, after the first year of treatment, we did get a significant decrease. And then also, again, after that 2023 treatment as well. All right, this is quick and dirty. So, fluoridone. I want to point out that failures are not true failures. We learn a lot from the science that we do. And if we keep on going and learn over time, we eventually get to a spot where we are being selective, and that is our main goal. Reducing applications over time, right? We're trying to understand and figure out the plant communities and under what water quality conditions are best candidates. And this way we're able to be more selective. We're working on additional questions right now, understanding, is there a difference between spring and fall applications? And a few non-target impacts were to coontail, elodea, and depending on the water body, we have observed some declines in naiad. Sorry, I'm jumping around here, but the third bullet is really important because even though we did find minimal impacts to native aquatic plants, this is not the case in all systems. If you have a system that is not very diverse, if you only have coontail and elodea, you're going to be knocking back those plants, and it also depends on water quality within your lake. So for Priscillacor, this was the first selective herbicide to provide multi-year control of hybrid water milfoil in a partial lake treatment in Minnesota. We did observe some impacts, like I said, to floating leaves, pondweeds, water shield, and we did get some good increases in native plants like clasping leaf pondweed, flat stem pondweed, stonewort, and water stargrass. For pinoxylum, we actually observed that considering pinoxylum has an extended exposure requirement, coupled with the observations in one of our other case studies, it's important to understand that aquatic plant managers should take into account inlets and outlets in their project, because we know from some of the research that we've done is that this can move off site quickly and you might not get control. There was variability in seasonal control, so you might want to do a larger scale treatment, much like a whole lake treatment. We observed that milfoil and curly leaf were managed within these treatment areas, so it could be considered for dual treatments. Our results, depending on if it was a spot treatment or a whole lake treatment, were kind of all over the place. But collectively, we did see reductions in sago pondweed and milfoil. Coontail and water stargrass were variable depending on the circumstance. And we did see some increases in white stem pondweed, water celery, macroalgae, elodea, and flat stem pondweed. With that, maybe I have time for questions.
Hi, April.
Hi.
I think we're going to go ahead with the next presenter just because we're trying to catch up on time. But if people have questions, please put them on the chat and they'll be able to answer them.
Sounds great.
And next up, we have Tina Fitzgerald. I was going to talk about the DNR detection and response plan. Take it away, Tina.
Awesome. You can hear me, see my screen?
Let's see your screen.
Awesome.
And hear you.
Awesome. Hi, everyone. Tina Fitzgerald with the DNR, talking about our early detection and response plan. And essentially what I wanted to tell everyone here today, especially since I know we have a few new local managers, is that a early detection and response plan exists for the state of Minnesota. You can find it on our website. If you go to our aquatic invasive species page, go to programs and reports, and scroll down to the early detection and response plan. So as we all know, early detection is key to finding infestations when we can potentially take management action or prevent further spread. So I would just encourage those that have not read this plan to read it. We also have, what's nice is a little flow chart here on how the detection and response process works here in Minnesota, because the DNR is tasked with confirming new infestations of aquatic invasive species in lakes and rivers in the state. This is the flow chart that our program uses to identify those. I like to explain it kind of as you look at a duck and it's just sitting on the water, but really underneath the feet are paddling wildly. And so that's what I kind of like to say about when we get a reported suspect aquatic invasive species to the DNR, there's a lot of moving parts that are happening that just may not be seen. And that's including verifying that report, assessing the population, assessing the risk, and communicating with partners. And so this flow chart is just a really helpful overview of how that process happens. So I encourage you to get familiar with it. And I also encourage you to reach out to your local aquatic invasive species specialist, a few of them you've heard from here today, to talk more about how response might happen in your local area. And honestly, with that, that's all I've got. So I'll pass it back to Doug and Rafa.
Thank you, Tina. Well, really appreciate that really important aspect of plant management here and rapid response. So I'm looking at the schedule, Rafa, and we're right at break. We're going to take break at 2:55. Maybe we should entertain, maybe save 5 minutes for questions and answers for our panelists. We'll be cutting into our break time a little bit.
Yep, I think that's a good idea.
Okay. Also, there are some questions going on in the chat, so I just want to alert you to that. Anybody have questions for our panelists or for our panelists for each other? Go ahead and take yourself off mic. I'm not seeing any in the chat. I'll ask a question for Chris. So I've heard that the bulbills, each bulbill can produce 50 plants. Have we observed that actually occurring here in Minnesota?
I have not heard of that. So, as far as I understand, a bulbil produces a plant. So I shouldn't say even plant, it's not a plant, it's macroalgae, but I don't know how many bulbils are attached to one plant. Each node can form a bulbil.
Right.
And then it's really hard to tease apart when you do pull one single plant up that what you're actually getting, but I'm just guessing, but if I could say how many bulbils I see attached, might be like 5. So, and then you have all your nodes, but unless somebody else knows the answer, I don't. It's challenging too, because they could be chopped up and there could cause more fragments in the water body, and then it's kind of multiplies even more. So that's all.
I had
Oh, go
a
ahead.
question for Christine.
Sure.
We did or we witnessed a copper sulfate on Starry Stoneware treatment, and we saw some burn down and then production of above bottom bulbils. And I'm just wondering if you have seen that in your area. And then with hand pulling, do you see a reduction in bubble production?
Your first question is, I guess it's a little fuzzy. The first paper out from Glisson et al. Did show that bulbils increased after copper treatment in the fall, but it's at the same time of year when you see bulbil production increase. So maybe. And I think just from what I've heard and talked to others, when you attack a plant, no matter if it's pesticide or hand pulling, it likes to like reproduce to keep alive, kind of survival mode. So it's, I don't know if there's any additional papers out there that can point that yes or no, but I would say likely it could, for your first question. And then what was your second question?
Oh, when we do hand pulling, if we find an individual plant that wasn't hand pulled earlier, it often has abundant bulbils, but it seems like when we re-pull later in the year, we just are finding a few undeveloped bulbils on those plants. So are you seeing a reduction of bulbils as you're hand pulling, or what's your feel on that?
Well, basically, if you're pulling the plants, you're getting as many bulbils that are attached, but there's a lot that remain in the sediment that is the reason why we're seeing regrowth. It's the bulbils in the sediment that aren't necessarily attached to the plant when you pull them. I forget for what your question was. I'm sorry again. But no, I would say it depends on when you're pulling the plants, because when you get towards fall, the bulbil production is a lot higher. So, but typically when you physically remove the starry stoneheart, you're removing the bulbils that are attached at the time, but there's definitely some that remain in the sediment.
Anybody else with questions? Not seeing any in the chat, Rafa?
No, none on the chat so far.
Yeah, thanks, Tina, for putting that resource in the chat. Go ahead and take yourself off mic if you have another question for any of our panelists. Hurry up and get it in. We got 1 minute to go. I have a question for April then. Well, we just hit 3 minutes, but maybe you can answer. How about how much does it cost per acre about for the chemicals that you were referring to earlier?
I don't know, to be
Well,
honest.
that's a quick answer then.
Yes, I feel like some of the county staff would have a better idea of that. I know galleon for sure is cost prohibitive. It's a new pesticide, and it's fairly expensive, and Priscillocor is definitely more expensive than some of the other ones, more generic ones like 2,4-D and triclopyr. Yeah, but maybe some of the county folks can give a ballpark.
Yep.
Yep.
So yeah, I see Alicia put in some information. We'll invite folks to during the break to put in if you do have any cost estimates for any of those 3 chemicals. So we are at 3:01. So we promised you a little bit of a break here. So how about if we reconvene at 3:06?
That sounds good.
Thanks, everybody. See you then.
Thank you.
Well, welcome back, everybody. I see that we are at 3:06. Maybe you can just give us a thumbs up to indicate that you're back here again. We can get going. All right. Seeing some thumbs up. All right, well, let's get going then with our third panel. And that is prioritizing management actions. And we're going to hear from Haley Olson, Alicia O'Hare, and Dan Sabulka. Haley, are you set? I will stop sharing my screen.
Yes, here.
Excellent.
All right, can you hear me?
Loud and clear.
Perfect. Well, hi, my name is Haley Olson, and I am a conservation technician at the Brown Soil and Water Conservation District located in Sleepy Eye, Minnesota. I recently started this position in January, so it's been a little bit over a year. We
Haley,
just
excuse me.
finished.
Could you turn on your camera? Are you able to do that?
Oh, can you not see me?
No.
Oh, mine shows that you can see me. Okay, let's see.
You can see Dan.
Okay, can you see me now?
Just 1 second. Still no.
Okay.
Did you have slides to share?
No, I did not.
Okay.
Well, I think that's fine, Doug. We can just keep it
Yeah.
going. Yeah.
Let's just keep it going then, Haley.
Okay, so we just finished our 5-year study on 4 of our priority lakes. So since we are located in southern Minnesota, we don't have very many lakes in our county. So we decided to prioritize AIS by doing a 5-year study on our 4 high value recreation lakes in our county. So we just wrapped that up last year. We did water quality monitoring, did some fisheries assessments, and some submerged aquatic vegetation surveys. So now we're currently in the process of working with LimnoPro to kind of figure out what our priorities should be for every lake and kind of go from there to make whatever management decisions we have to make. So that's kind of what we've been focusing so far with our management priorities with the limited number of lakes that we have in our county.
Thank you. And if Dan or Alicia, if you want to go next.
Ladies first.
I guess I can't argue with that, can I? I'm Alicia O'Hare. I'm the water resource specialist at Right Soil and Water. So in addition to AIS, I do a lot of the water monitoring and stuff. Just to say, I have to prioritize AIS all the time, just in how much time I spend. But we established a AIS committee in 2019 following the regional inspection program pilot that took place in 2018 to help guide us through the differences of opinion of some of the approaches to AIS. So now we have that specific committee that meets 4 times a year and can take a deeper dive into some of the AIS issues, like how are we structuring our treatment grant, where are the inspectors going for that, and then we can have a committee talk about those different things. So that's definitely one of the ways that we prioritize our AIS things. And we just redid our AIS management plan last year, more in the style of the way watershed-based implementation is done, with the actions and goals and that sort of thing.
Thank you. And Dan.
Hi, everyone. Dan Sabulka. I'm the Senior Water Resource Specialist with Sherburne Soil and Water Conservation District. And so, similar to Alicia, I oversee a lot of our watershed planning projects, but also oversee our aquatic invasive species prevention program on behalf of Sherburne County. within Sherburne County, we've got a little over 100 lakes. I would say 18 of which are larger, more prominent public access lakes where we have watercraft and trailers, coming and going from. So not nearly as many as some of the other counties, perhaps in the northern part of the state, but enough to have some fun with. And at that level, there's comes a certain amount of prioritizing not only by water body, but also by the activities that are conducted within our AIS prevention program. And by that, I mean, our priorities be more along the lines of education or prevention or containment of existing species that are here. And so I think that's something that everybody, should both consider, but also adapt to as things inevitably change, is what is the setting that you are working within for your county landscape. Like Alicia, we also established a aquatic invasive species task force or committee, if you will, early on when we started getting the prevention aid, so around 2015. And this task force has been active since then. We develop a 5-year plan every 5 years. And so they get a little bit more face time and more conversations occur in developing those plans. But while the plans are in progress, then there are times where we get together to, as for an annual summary meeting or presentation that's coming along. But the task force itself, I think, is a great way to help in priority setting and goal setting. With our task force, we've strived to try and include some differing perspectives and voices from the community. So Lake Association or resident folks are obviously involved in that group, trying to get anglers or Angler Association members to be a part of that group. We have county, city, township representatives that are active in the group. Lastly, I suppose we have had 1 or 2 people that have been involved in the watercraft or water accessories kind of industry, if you will, just again to bring a different perspective and opinions to that group.
Okay, thank you for that introduction from all of you, and it's great to have you here. Our time is going to go by pretty fast, so I'm going to go ahead and just go straight into our questions. So in a world of limited resources, how do you prioritize AIS activities? And any of you can start.
I think there's 2 major limitations for us, and those are budget and time. It's like there's no shortage of ideas out there. Like one of our biggest things with the AIS's committee is they have, hey, we should do this and we should do that. And I go, okay, where do you want the money to come from? So we really have to think back to like how much money do we have to work with, and where do you want to put it generally between inspection and treatment. And then our like tool station, we just did like some low cost tool stations and that sort of thing. So it's just one of those things of, having the committee that can do the deeper dive and talk about the ideas. But then like they're actually not a decision-making body. They're a committee. Our board still has that final say. So it's good to have the ideas machine and like okay, let's look deeper into this, but then having the actual board of supervisors give the final okay on the ideas that we've really, filtered out has been really helpful in figuring that out.
Yeah, so to follow that, it's kind of the same thing that we're dealing with is staff time and also budgeting. These last couple of years, we haven't done much for management since we finished our 5-year plan, and we've been trying to put away some funding, hoping that this year we'll be able to use a lot of that funding towards management actions once we have our priorities set. Kind of like Alicia said, we have to go through with our priorities through our board. So they're kind of a main factor in helping us determine what we should focus on. We have curly leaf pondweed currently in one of our high value lakes. So I know that that's probably going to be a priority for us this next season. This summer we did install aquatic weed stick removal station. So that's been a good first step for us in kind of prioritizing and getting the word out to the public. So we're just kind of going through there, working with our board to figure out what areas for each lake that we want to prioritize for different management strategies this next season.
And I would say, just to add on to those great points, you know what, when you're developing priorities for something like aquatic invasive species or anything else, you know, it helps to identify who is doing the priority setting. So Alicia and Haley both mentioned that their boards ultimately make the call, but there might be a committee or something that provides some input. And so, you know, those committees are great to lean upon because it grabs, it helps to develop buy-in from the community. If it's just myself that's coming up with priorities and I pitch that to the board, then you know, I may or may not have a good take on what the community as a whole values as far as our AIS program. And so having a committee that is involved in those conversations and really facilitating the discussion well, I think is an important point. You know, so having a committee that is knowledgeable to a certain extent about the issues and that you can ask the right questions, have the right activities, you know, putting a colored sticky note, a red or a blue or a green sticky note up on a poster or something like that. There's a lot of different ways to engage a group and to get feedback on their priorities, but, you know, facilitating that conversation and having those different activities really helps to bring things to the forefront that are more important than other things, if that makes sense. And so, you know, that's important for a number of reasons, because when you get that information, you incorporate that into your plan, but you can document why it's important, because ultimately there will be a point in time where the plan may need to change. You may be questioned by somebody from the public about why this activity was done. Why are you focusing so much on education and not enough on watercraft inspections? Well, this was a priority as demonstrated by our committee, who showed strong support with 72% of committee members committing to that area versus the other area. Those kind of things are really important to document and include in some type of a strategic plan. And again, having that type of experience with a committee and getting that kind of information just comforts you to know that you have the community buy-in for your program.
Thank you all for those great answers. And so I've been hearing from all of you, I've heard that you have to get your committee involved, you have to get your board involved. But this next question is kind of going to add a little bit to that. And this one's talking more about stakeholders. So how do you engage local stakeholders in prioritization conversations and decisions? And I'm also sharing this on the chat if you just want to take a look at it as well.
When we updated our AIS management plan, we did send out a survey to the public. We got about 60 responses, if I'm remembering my numbers correctly. But so we posted it online, we had it available, and then we also had it open for comments as well. None of this was required. The AIS management plan doesn't have the strict rules that a watershed-based plan has in order to, for those comments, but we wanted to make sure that we heard those comments, so we did make it available. Our AIS committee is, we hope, made up of those stakeholders. We have 2 SWCD supervisors, 2 county commissioners. We have 2 lake association members, but only one of them has voting rights. We have 2 sportsmen, again, only one with voting rights, but 2 for the purposes of discussion, and another elected official as all the seats on our AIS committee. So we keep those open, and we ask for a recommendation from some type of body. So it's not enough just to have a guy say, hey, I fish, I want to be your sportsman representative. We try to have a entity behind them, like the West Metro Anglers Association or Bass, to recommend them to our committee so that there is local support.
Yeah, so Alicia, I kind of like your idea of sending out the survey to people. We haven't gotten to the point yet where we're able to reach out to the public yet, but I like that idea. But we also kind of have a plan of potentially meeting. We have a couple of smaller lake associations, so kind of reaching out with them, and then sportsmen's clubs to kind of see what they think their priorities are and kind of hear what they have to say about it, and just take what they have into consideration. Like I said, we don't have a whole lot of active lake associations, but kind of just reaching out to the ones we know that they know we just finished our 5-year study and kind of see what they want us to primarily focus on.
Yeah, we also sent out a stakeholder survey and an anonymous electronic stakeholder survey. And one of the questions I included on there was, one of the first couple of questions was something along the lines of, what is your level of knowledge pertaining to aquatic invasive species? Which is important, because when you conduct those public surveys, you don't know what you're going to get. You may have experts, you may have people that think AIS stands for artificial intelligence systems or something, or anything in between. So you take those results and I coupled them with our AIS task force conversations to create a holistic picture of what the perspectives are. And engaging folks to be on the task force in the first place, that can be a challenge. And like Alicia said, a great idea is to reach out to a group and ask that they have a liaison that commits to being on that task force. Reach out to somebody directly with a phone call or stop by their office. Sometimes an email it's too easy to ignore. So to ask of people to spend an evening or time away from their lives to commit to your task force it can be a large ask sometimes. But if you do that directly, I've gotten a better response that way.
Hey, thank you for that. And so this year we have a lot of new managers. It seems we just got that from a survey. So I really want to get to this question, and I'm posting it on the chat so you can read it. And it is, what advice do you have for those interested in starting a prioritization effort?
Think about what success means in your prioritization effort. And it really depends. Some of it's easy, some of it's not. Like, treatment is hard. In what is success in a treatment grant program? Just when you want to keep it easy, it sometimes is just, here's the entities that we're helping out. Here's the different species that we're looking at. But have those measures of success. When we look at our inspection program, we have made our measures of success not only number of inspections per hour, but the number of license plates that we look at. Because if you're inspecting the same people over and over and over again, you're doing a lot of inspections, but eventually it kind of falls on deaf ears. So you want to make sure that you are inspecting a wide range of people. So incorporating both of those measures of success and keeping the public informed of those measures has been really useful, especially when it relates to our inspection program, which is the largest portion of our AIS budget. So.
To add to that, I would say to kind of look at what funding you have available. That can make a big difference on what future projects you may or may not be able to do. And with all the 1W, 1P funding, that's kind of been a priority for us, is to look at that plan and see what we could possibly do for management actions or in the future to see what we could potentially fund. So just remember to look at what funding you have available that might be able to help you figure out what areas to prioritize.
Yeah, I guess I would just add, define who it is that's doing the prioritization. Who is it? Is it a board? Is it a council? Is it a task force or committee? And then, identify what types of activities are going to best get the information you need from that group. And that's where some of the, I mentioned earlier, uh, meeting facilitation and different exercises to get information out of people. That's good to have in your back pocket. We were very fortunate. We sat down with our AIS task force and we could have easily turned a 2-hour meeting into a 6-hour meeting because everybody loves to chat about AIS, but that's not always the case. I've facilitated meetings where it's hard to get somebody to raise their hand. It's hard for somebody to speak up. And so if your decision is that there is a body of people that are going to be communicating priorities, you need some type of mechanism to get that information. And maybe it is an anonymous survey. Maybe it's an exercise where everybody gets up and puts different sticky notes on poster board. There's a lot of different ways to do that. And so do some research, look at your neighboring counties, the plans that they've put together, and do they identify some of those things within their plans? A lot of resources out there for this type of work.
Okay, thank you. And so we have about 3 minutes left. Is there anybody in the audience that has some burning questions here for your colleagues? Again, this is the panel for prioritizing management actions. So anybody have any questions? Maybe there's something that you want to try out. Maybe there's something that you've heard that other counties are doing, or maybe just sharing some difficulties that you have. This is kind of the time and the space to start to talk about that. So. Okay, so Drew, you can unmute yourself.
All right. Yeah, I guess question for panelists or whoever wants to answer. Are you, Alicia, Dan, Haley, seeing any shifts in your AIS priority, I guess, sentiments coming from stakeholders or task force members? And what I mean by that is, and I can use ourselves as an example, due to, both limited funding and limited ability to pull in the amount of staff that we would need for watercraft inspections, we're seeing some of our resources sort of redefined or reprioritized to other AIS activities. And I'm just wondering if that's been a trend in your organizations or in your areas as well.
Yeah, and if you can keep it to about 30-second responses so we can get through all of them. Thank you.
Yeah, I'll jump in. That's a great question. In a recent example would be that for years we've had, within the category of education, we've had a lot of education activities that were geared towards people that live on lakes and lake association members. In our last AIS task force, when we sat down, we said, you know what, we're perhaps focusing on that a little too much. We should be focusing on the people that are transient boaters. They visit a lake 2-3 times a year. And so doing a variety of education to capture those people that are not as engaged with this topic would be preferable. And so for the third edition of our plan, we're placing a little more focus on billboards, on tabling events at farmers markets, things of that nature, to view those occasional lake visitors.
We had to do a lot of prioritization because when I started in 2016, our going rate for AIS inspections was about $15 an hour. In 2024, we were paying $24 an hour. So it like big shift in just how much can you do fast. And we had to make those choices, and we found that we weren't doing weekdays anymore, not in the morning. We were doing them in the evening, and then we were getting better results there. And our most recent shift is in education. We're going to try and engage the high school fishing teams a little bit more and do some education around there. We had been previously doing more of the billboards and signs and things, and we're going to shift back to the more intimate small group education.
I would say we're doing the same thing as Alicia. We're trying to focus more on in-person interactions and trying to do some classroom presentations with high school ages and trying to have a presence at youth fishing events and just things like that, trying to engage the younger audience. Because we currently do a lot of digital ads, so we're kind of reaching that older audience. So trying to just find a different target.
Okay. Well, thank you all so much for your time. And I think we're gonna continue on to stay on time. And that was for some great responses. And I wish we could just keep talking about all of this, but we have to keep going. Thank you.
Yes, thank you to all of our panelists. That was great. We're on to our next and final panel, connecting AIS and water quality. And we're going to get a double dose from a couple of folks that have already spoken, including Dan and Justin and Andrew, and also welcome to Amanda. So Dan, if you want to kick it off.
Hey everyone,
also,
Dan Smith.
I just want to mention
Go
there
ahead.
are some comments in the chat, so I just wanted to draw everybody's attention to that. If you have any comments for discussion, please do so. Thanks.
Yeah.
Go ahead, Dan.
Great to be here again talking about this topic. As I mentioned earlier, with Sherburne Soil and Water, a lot of the work that I do is both in the aquatic invasive species realm, but also overseeing watershed plans and projects and water quality projects for our county too. And those two worlds collide quite a bit, I would say. There's a lot of documented history about aquatic invasive species and impacts on water quality, but also the ecological health of a lake as well. So happy to be here discussing that today. I think we're just doing introductions right now, or?
Yeah, okay. We're just doing introductions, yes. All right, so Amanda.
Hi, Amanda. I'm the AIS supervisor coordinator person for Cook County. I'm in Grand Marais, Minnesota. I've been doing this for 10 years. Thanks for having me.
Okay, and then we'll go with Drew.
All right, yeah. So again, Drew Dickhart, Carver County AIS coordinator. And this is my, I believe, 9th year here in this role with the county. And this is a great topic. I'm glad it was chosen, and yeah, should be some good discussion.
Okay, and then we have Justin.
Yep, Justin again, Ramsey County. I believe this is my 7th or 8th year. Yeah, a little bit shorter timer.
All right, well, thank you all for those introductions, and we're going to go right into it. So we're talking about water quality, and here we're working with aquatic invasive species. So the first question that we have for you all is, what connections has your program made between AIS and water quality initiatives? And again, like in the last panel, I will put that in the chat so you can read it. And anybody can start.
I will start. I don't know if this is what the panel was meant for, but for water quality, for us, we're near the boundary waters, and so our water quality is one of the best in the state. And our Soil and Water has had a long partnership with lake monitors and volunteers to do water quality sampling through the DNR Sentinel Lakes or for MPCA. And so when AIS became established in the county, it was easy for me to pool those already established water monitors for monitoring for invasive species at the time. So they're already going out every week or every month to do all these other water quality parameters, and then on that, finding water food net or crayfish traps for them.
I can mention a little bit. I think I might be a little bit unique in some of these groups I'm finding is that because the other half of my job is terrestrial invasives and not water quality. We have a whole different water quality team, and we're obviously highly urban, surrounded by watershed districts, watershed management organizations. So they're all taking care of a lot of that, where we've been able to help supplement a little bit is find those niches in like curlyleaf pondweed or phragmites or doing delineations for potential treatments that might have otherwise been contracted out or to some engineering companies or something like that. Being able to work more closely with the watershed districts to bring in the water quality component to that, or to the invasive species that they might have not have otherwise been looking at. And like for instance, I do basically nothing with carp because watershed districts have that covered. That's a big water quality component. They take care of it all. So it's been a little bit more on the researching how what the next level of involvement in AIS and its relationship to water quality. So for instance, replantings. We've done a couple of the replantings post-treatment because the watershed districts have the capacity and the knowledge on the treatment aspect of it. But what we haven't been able to do is replanting natives in the absence of non-native. So we've gotten involved with a little bit of that.
This is Andrew. I'm in maybe sort of the opposite situation as Justin. So my department is the planning and water management department. So within our department is our water quality team. So we really do try to stress that it's established, right, that there are interconnections between aquatic invasive species and water quality. We know this already. And I think what we are trying to do is not, we know there are relationships, but we want to try to better understand these relationships. And we also want to incorporate AIS monitoring in with other water quality monitoring. They don't need to be separate adventures, right? You're going out in a boat on a lake, try to incorporate that together if you have the capability to. And we have, summer interns that can help us do that. And then also in our reporting too, right? We don't want, maybe this is just my opinion, but I would like our readers, our community who is active within our, what we do in our department. I don't want them to think that AIS management is completely distinct from water quality management, because it really is not. They really do go together. And, I could share a few examples, but maybe I'll let someone else kind of chime in here and then come back. But I think it's really important, especially in having our discussions and reporting about AIS, that we keep that connection to water quality kind of at the surface, so to speak. Thanks.
Yes, some of the work that I've done in connecting AIS with water quality, to a limited extent, I've done some environmental modeling or monitoring rather on waterways that are below wetland systems. We often think of wetlands as being the liver, the kidney of the environment, taking in pollution and emitting out cleaner water, and that's not always the case. And there's been a couple circumstances that we've monitored water downstream of hybrid cattail dominated wetlands. And when you have a monoculture of cattail, a hybrid cattail that has a large oxygen demand. You can get anaerobic conditions in those wetlands and a release of dissolved phosphorus, which is the type that's more readily available to algae. And so that's something that I've not to the level of researching, but monitoring and trying to understand wetlands. Are they a source or are they a sink as far as phosphorus is concerned to downstream water bodies? That's something I've dove into a little bit. I know there's a researcher, Amy Schrank, I believe, with MACERC that's been doing some work with cattail monocultures on a lake scale or lakeshore area and implications on dissolved oxygen. So there's more work to be done there, I think, in connecting those two. The other work that I've done has been with carp. So there's a couple of lakes that we've been monitoring carp, assessing the carp population. There's a recommended management threshold of 89 kilograms per hectare, I believe it is, or about 100 pounds per acre. After you exceed that amount of biomass of carp in a system, then you start to see water quality impacts with turbid water, with a decline of aquatic plants, et cetera. So some of the work that I'm doing, with a few different lake groups is having those initial surveys take place to understand what is the carp population, what is the biomass amount, because that's not information, unfortunately, that you can get from a DNR survey of the lake when they're doing a fisheries assessment. They're typically looking for the game fish species, the walleye, the perch, the bass, et cetera. So a separately designed survey to look at carp is what's necessary. And then management of carp is going to improve the water quality on the system. There's some research out there that suggests that carp can, through their stirring up the bottom, through that kind of activity, they can produce a couple of pounds of phosphorus per acre on a lake scale. Also, the fish themselves can be quite big. I've seen another report where you can use an estimate of 0.05% of a carp's biomass is phosphorus. So for one lake in particular that I'm working on right now with our Lab to Lakes project, which is a project that's funded through MACERC, with the estimates that we have of carp in that lake, if we were to bring the carp population down to that management threshold, then we would be removing about 140 pounds of phosphorus from that lake just by taking the fish out alone. So that's a substantial reduction in the phosphorus budget just through removing the fish, but let alone these fish also produce more phosphorus to the water column by stirring up the bottom. So there's a very strong connection with that particular species.
Yeah, thank you. That helped. Thank you all for those responses. And I do want to keep in mind that we have a lot of new managers this year, it seems like. So something they might be looking at, how can they start making these partnerships, or like, who do I talk to start connecting water quality with aquatic invasive species? So I don't know why I keep getting muted. I think I'm pressing the enter key and I had it right there. So apologies. So the next question is, who are the partners and stakeholders you have engaged in this work?
The partners that I have engaged in this work are the lake associations, the people that live on the lakes, even if they're not a part of a lake association, the resorts and outfitters that are on the lakes. The University of Minnesota Extension also helps out quite a bit. They're very useful. And it's a lot of networking, kind of like your business proposal. Like, I don't know if you're a used car salesman, but like you need to go and make those connections and keep calling them and finding that common ground, which is having good quality water. And since they live on the lake, it's their lake property, their home property values would be benefited if they keep clean water.
I'd say you have one watershed district or watershed management organization. If you don't, or if you're not the soil and water, contact your soil and water. Definitely the associations. There's a lot that can be done with the homeowners as well, just connecting that whole piece of what you do on your shoreline effects and what you have on your boat effects, and just kind of doing some of that general education with what you do matters. So keeping it simple of, hey, your clean boat, yeah, that's good. Hey, your shoreline's clean of invasive species and has some diversity to it. Hey, that's good stuff. That's what we want. Keeping that messaging nice and clear from the upland all the way down into the water has been helpful for us as we partner with other organizations.
I can speak a little on both the partnerships and kind of connections made. So Dan brought up a great point with the carp. I was going to talk about that a little bit too. So in Carver County, we have a carp management project. It's been going on for 8 years or more now. And we also have a relatively new goldfish project where we have a chain of lakes that got infested with gazillions of goldfish. And one of the connections here with the goldfish is that, you know, for carp, we have that research, we have that management threshold, right, of 89, kilograms per hectare or 100 pounds per acre. We don't have that with goldfish, right? So we hopefully can use some of our management activities and hopefully others together to try to come up with that threshold. Is it going to, right now we're just assuming that it's going to be similar to carp, but we don't know that. So as we continue to find ways to remove goldfish from the system, we're going to try to, you know, track those water quality metrics, maybe even more than we do in our normal water quality monitoring routine, pay attention to maybe some other things more closely. Let's see, what else? So zebra mussels, kind of another example, kind of opposite of carp, right? So carp and goldfish are going to resuspend sediment up in to the water column, which can then give you, you know, algal blooms and high nutrients. Zebra mussels, on the other hand, are going to filter out those suspended particles, right? And they're going to bring those nutrients to the bottom. And then you're going to see better competition of other planktonic species in the water column. And then blue-green algae and other toxic algal blooms are going to kind of dominate that system. So, I mean, these are just examples of connections that we already know about. And with the goldfish, again, I hope that we can sort of continue making some connections that we can use. I guess we can bring that data-driven research, what am I trying to say, into actual management activities.
Yeah, I think there's been a lot of great, stakeholder groups brought forward by the others here. The only thing I would add, I think we covered MACERC, U of M. They're obviously doing a lot of like great pioneering research that you should be aware of and incorporating into your programs if it's suitable. The other, as far as other stakeholders are concerned, I would really encourage everybody to reach out to cities if the lake that you're working on is within a city limits. They often are aware of what's going on with the lake and understand that it is an asset for the community, so have a vested interest in helping to monitor, improve the conditions, etc. So that can be a stakeholder group that you can engage with. Similarly for townships, if there's a township that is appropriate for you to reach out to and work with, or maybe they just want to be, kept aware of what's happening with the management of that resource that again is an asset to the communities. So lake associations as well, but some of those some of those other municipalities that might be interested in learning more about the lake or stream, whatever you're working on.
I forgot to talk about partnerships. I was actually going to use the Goldfish Project as an example. So that the Goldfish Project is a chain of lakes in Chaska, and a lot of the lakes that we're doing the management in have paved city-owned trails around them. So the community can easily see what's going on. They see the goldfish, they post about it, they call us sometimes to see what we're doing. So we use that to our advantage. And there are some folks who either run or cycle or walk around these trails almost daily. And we kind of established relationship with some of them. And they're kind of our sentinels when they see the goldfish moving from one area of the lake over to like shallow inlet to when it looks like they're aggregating. We have them give us a call. We actually have like a group text that we all use, and the volunteers will just kind of let us know, hey guys, we're seeing the goldfish over near the blah 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 aggregating, and that's a great way for us to know, okay, we need to figure out how we're gonna respond to this. Are we going to do a removal? Do we have staff available? Blah blah blah. So that's been a great partnership. There's not really a lake association, so to speak, around these lakes. So it's more of an informal kind of community relationship. And it's been working great so far.
Okay, thank you. I unmuted myself that time. So we have about 3 minutes left. Now I'm going to open up to the audience in case you all have any questions. As Doug mentioned on the chat, on this panel right here, we have over 20 years of experience. So this is the time to ask those questions, especially if you're new. Yeah, so any questions?
Maybe I didn't do my math correct. It's probably closer to 30.
Yeah, I think it's more than that, Doug. Right.
I'm not seeing any questions in the chat.
Okay.
Well, while we're waiting, you had the question on, what did it say? How do you get into water quality? What was the last question? Sorry, I'm looking at it. If you're interested in doing something similar. Yeah. What advice for those who wanted? So if you want to start connecting water quality, my first thought was start with the gateway plants like Phragmites and flowering rush and curly pondweed. They're big, they're visible, they have a very direct and documented impact. So it's really simple to get into those and say, well, I'm going to help treat or provide some technical assistance to people treating Phragmites because it can invade wetlands and respiration and cause water tables to go all funky. So that's an easy one, I think, for people to get into. And then obviously curly because of it senescing and causing phosphorus and all that. If you can even get in on some education pieces around that to start to educate people that's why it's a problem. It's a really easy one to dip your toes into.
Okay. And yeah, we're going to continue with that one. Do you all have anything to add to that? Any advice that you have for those that are interested in doing something similar? And you all are doing really different things. So that will be something good to add. His final words. We're right on time.
Yes, we are. We're right on time. Just want to thank again all of our panelists today. Everybody did an excellent job. Hopefully, all of you that attended learned and enjoyed the format that we had in terms of panels. You said in your surveys that you'd like to have more panels, so we listened and we put together these different panels. So hopefully you got a lot out of them. I know I did. So just want to thank all of you, all the county folks, our partners, for joining us. Also want to recognize we had a good chunk of DNR folks also listening in today. So they're here also to support you. Rafa and I are here to support you, Tina, as well. So if you have any needs, be sure to reach out for us. That's what we're here for. So with that, I think I'm just going to show briefly again the coming up workshops reminder. So our next one is on February 20th from 9 till noon. And then we have the public engagement workshop March 25th, 1 to 4, and then our 2 in-person workshops in the south and in the north. So put those on your calendar, and we hope to see you at the upcoming workshops. Rafa, do you have anything else to add?
Yeah, no, that's all I have. For now, the partnerships workshop and the engagement workshops, we asked you to put a hold on that. This week, we are going to send an invite out for a Teams invite, just like this one. So look for that one this week.
Thanks, Rafa. I think we can call this workshop adjourned, huh?
Yeah. Thank you, everybody.
Thanks all.